Thanks again, Ben, for a wonderful introduction. I really feel like I kind of sort of tricked people into this presentation because there's no autism in the title, but a lot of the talk will be about autism, given that I'm in the Autism Research Center. Keith and Renee already gave a wonderful introduction on oxytocin in general, but I still like to sort of go back to the basics, especially when it relates to autism, because this is sort of like around the time where I started into this field. These studies started coming out on oxytocin improving social cognition in people with autism in very sort of select studies. And I think this really created like a massive hype as oxytocin being a having potential therapeutic value in autism. And that spurred like a whole sort of side industry, so it seemed, and a lot of these things are still around. Like these are the ones that I could find this morning, still on the internet. The liquid trust one, I think you can still buy off Amazon. I don't think, it's not a nasal spray, I think it's just like a sort of a pheromone or something you're supposed to spray on yourself, and then people will trust you or like you. I haven't tried that one, I've tried the other one. It says it's clinically proven, so it must work. I don't know of any clinical trials that actually support this study to do anything in autism, but the first clinical trials for oxytocin in autism are starting to come out. But I don't think you need to join the Live Love Club today to actually take part in those. You can just go to the clinicaltrials.gov website and see where those trials are happening. Now, this hype it's been good and bad. So the bad side is it's spurred of all these like sort of commercial enterprises trying to sell people oxytocin in various forms, whether it's nasal spray, drops, pheromone sprays. The positive thing about it is that it also spurred on a lot of research, and it sort of basically gave all of us here a career, more or less. And there have been quite a few studies that have supported these sort of pro-social positive effects of oxytocin. I've listed a few here. Keith's mentioned quite a few. Renee's mentioned quite a few. I think now in the past 2-3 years, there's some sort of meta-analyses starting to come out, both in clinical populations as well as just across different cognitive domains. When I put up this slide, I always need to like also put a strong caveat. It's not just all pro-social and happy and positive. There's also some sort of negative effects of oxytocin depending on the specific context that it's administered in. Now, if we kind of go back, though, because we've done all these studies, but we're still kind of struggling with this question, what is oxytocin really and what does it do? At OHBM, I think like when you look at this picture where we kind of start, is we start in the middle when we try to go out, because we're interested in the brain, and then we try and sort of infer behavior from that, or we try and relate behavior to that. Or we try and sort of, after we do all the fancy imaging, we try and sort of go back to the biology and try and understand what's actually driving these imaging findings. But I think this is a sort of also a nice framework to think about how to study this clinically. Like on the one hand, you can sort of study this at a behavioral level, like a more phenotypic element, and on the other hand, it's like the more sort of molecular biology, if you were. So although this is an imaging conference, I kind of like, I started out on the behavioral level and then sort of transitioned into imaging, and I'm trying to do the reverse thing. So the very first study I did with oxytocin was in individuals with autism. And I do always sort of start with this slide because it kind of re-emphasizes this point, is that it's not just all positive. So what we did in this study is we administered oxytocin to men with and without autism. And then we did a clinical interview where we kind of eye-tracked to see how much eye contact they were actually making with the clinical interview, like during the clinical interview with the clinician. What's nice about this study is I think initially when we looked at these results, we didn't find any strong effects, despite there being lots of studies that showed if you give someone oxytocin, they're going to make more eye contact. Like this sort of social salience network perhaps is what might underlie this and what sort of like reinforces this people making eye contact. Initially when we looked at this study, we didn't find this. We just did like the sort of standard like group comparison, case control kind of design. But then when we sort of delved into it a little bit further, we could actually find that the individuals that already made like a decent amount of eye contact to begin with, they just didn't improve as much. Whereas the individuals that were sort of most severely affected to begin with, those were the ones that actually showed some improvement. So this kind of like goes back to this, it's kind of context and individual specific, what Keith already mentioned as well. Now sort of going to the other side of that first picture where like you have behavior on one side, but also there's biology underlying all of this, and like sort of trying to sort of delve into this, there have been different hypotheses as to like how is this oxytocin system sort of organized in the brain. I think there's quite a lot of these different hypotheses. I'm not going to go into much detail. I think what all of these three sort of, they're not mutually exclusive either, but I think what they all sort of emphasize is that there's actually very little sort of temporal and spatial specificity in the oxytocin system in the brain. And when you sort of look at all these different biological underpinnings, it kind of seems like it might be some sort of like Wi-Fi signal, and it has the potential to reach basically every region in the brain, depending on the context, but depending on the sort of mechanism, the specific mechanism of action. So although this is very nice, and it kind of like gives this potential for oxytocin to influence the brain in a very sort of broad way, it doesn't give us a lot of specificity. So you can also look at the other end of that biology and try and figure out like where are these oxytocin receptors in the brain actually located, because ultimately that's going to be the target of, if you're administering oxytocin, you kind of assume it's triggering these receptors, and these receptors are then causing some cascade that ultimately leads to a change in behavior. I'm not sure if you can see it very well on this slide. This is all post-mortem gene expression data that's publicly available, and I definitely encourage people to sort of delve into this data further. And more spatially detailed. This is just sort of looking like, are there specific regions in the brain where we have higher densities of oxytocin receptors? Because up until I think like a couple of years ago, the only evidence we had of this came from animal studies, and even in animal studies, we kind of know that these receptor densities vary like even within species. So it's very sort of hard to pinpoint where you'd exactly expect effects. And I think a lot of the, as Renee already mentioned, a lot of the human literature is sort of focused on amygdala. And I was very surprised when I first sort of created this plot as a sort of quantitative, to try and sort of quantify like where these receptors were, is that the amygdala is one of the subcortical regions that, at least in post-mortem gene expression, shows one of the lowest levels of oxytocin receptor. Now I'm sure there's some caveats because it's all post-mortem, so I'm sure there's also changes in how these receptors are expressed over age. Again, this just shows it's a very diffuse system, so it still remains quite tricky to actually predict where we're going to find effects on the brain. So the next step, and this is roughly where I come in, and I think more or less the reason why I'm in this symposium, is I tried to sort of review these studies, and I think I was very lucky when I did this review, because by now there's so many resting state and connectivity studies on oxytocin. I think when I started there was one resting state study on oxytocin, so it was very easy to do a review. There were other connectivity studies at the time, but they were all like in a specific context or like during a specific task in the scanner. And if you sort of just glance over this literature, you can kind of see that depending on the context, depending on the task, you find connectivity effects, but they're definitely context dependent. So when you do something like resting state, or you want to get like a more baseline idea of how does administering oxytocin actually affect this system, this might not be the most informative way to go about it either. So instead, we tried to sort of get a slightly more data-driven, open-minded approach to the whole thing. We did resting state fMRI using multi-echo resting state scanning. I only put in the resting state here, but we also have data from loads of tasks, which I'm not going to present here. The other thing I should mention is my review paper. What I found when I did that review is nearly like, in general, probably in neuroscience, most of the literature is very male-biased. I think now in the last 2 to 3 years, Renee is one of these people, and there's more and more studies on females and how they respond to oxytocin, but definitely 5-6 years ago, I think 95% of the literature was on men. And we know also that there's definitely some sort of interaction with sex. So we studied women with and without autism. I'm initially going to show you some of the data from the women without autism. So taking a very sort of data-driven approach, we ran ICA analysis to identify all these different networks that you get during resting state. And then we basically looked at all the possible combinations of all these different networks, and not assuming that there was going to be any specific network that was going to be affected by oxytocin, just see like which of these networks actually show changes. And across all these different comparisons, I think if I remember correctly, we had about 30 ICA components, so roughly 30 networks. And we just compared all of them and then corrected for all the multiple comparisons, and there was only one component pair that actually stood out. So as you can see in panels A and B, these are sort of the two components where we found an effect of oxytocin. And then again, trying to sort of keep it very data-driven, we ran these components through NeuroSynth to get an idea of what their sort of functional meaning might be based on this automated meta-analysis. And we find that, as we might have sort of expected based on previous literature, but again, we didn't make any assumptions, we find that one of these networks is clearly sort of a reward network. It sort of strongly overlaps with striatal regions. There's some amygdala in that component as well. And then the other network is sort of more of a cortical region involved in pain processing and auditory processing. So this is, in a way, it's sort of a socio-emotional reward network, perhaps, if you can call it that broadly. And then, as you can see in panel E, I don't think the lines are very clear, but every data point in that graph is one subject. And then for every subject, we can basically see do they increase their connectivity between these two networks when we give them oxytocin or not. And this effect really stood out as very robust. We ended up with 27 women in this study. And out of the 27, 26 showed an effect in the same direction, and one person showed no change. And this person is also the one that's the most extreme outlier in the placebo condition. So a really strong effect. And then, obviously, because we're interested in autism and how does this sort of feed back into autism, we also checked to see whether this effect was related to their autistic traits. So that's the right graph, the lower right graph you can see. And we see that this sort of neatly scales. Like the individuals that had higher autistic traits also showed a stronger increase in connectivity after we gave them oxytocin. Now, I think when I got to this stage, I was very happy. I thought, well, I'm just going to write the paper and we're done, because this is an extremely nice effect. Luckily, I also had one co-author, Mike Lombardo, who was much more skeptical of this stuff than I was at the time. Who was like, but how sure can you be that your placebo condition is a good placebo condition? Because you're still putting these people through like all of the scanning, you're administering stuff. So how representative is that of an actual like normal scanning session? Now, we're also lucky that at the same time, other people were scanning on the same scanner with the same protocol, and they were willing to just give us their data for free, so we could do like a sort of out-of-group, again, validation of this network. So we ran the exact same protocol, the exact same ICA analysis in this group. And we identified two components that show extremely high spatial overlap with the two components where we found an effect, and then just check to see whether that baseline connectivity is essentially zero, because that's what we see in our placebo condition. And we confirmed that actually our placebo condition very nicely sort of mimics the normal system. So then sort of moving on from this is all in healthy volunteers, people without a diagnosis. So next we kind of want to know, like, what does this do in autism? And sort of based on this panel E figure, we had expected, well, you get people with autism in, their autistic traits are going to be off the chart, so surely they're going to show a massive effect of oxytocin when you look at functional connectivity. So this is, again, running the exact same analysis, comparing between all the different ICA components. It's kind of hard to see, we do identify 3 pairs of components in this analysis. That's the first thing that's surprising, is we get more effect somehow. The other thing I should notice is that the effects in this group are much smaller. So the largest effect size I think we found in this group is a Cohen's d of 0.6 or 0.7, whereas in our original finding, the Cohen's d was, I think, 1.4 or 1.5, quite high. The other thing that's interesting to note is that actually the components that we find are not identical to the ones we found in our healthy volunteer group. We do still have the subcortical component that largely overlaps with striatum and shows, again, high sort of spatial correlation with the component from the previous analysis. But then the sort of more cortical components actually seem to resemble default mode network kind of components. So that's sort of the first interesting element to notice about this. The other thing is you can't really see it in these figures, but it's definitely not the case that all the individuals with autism improve. Some declines. It's much more heterogeneous. It's almost evenly split in our autism group. Now, of course, we also wanted to see, like, but what if we actually like get these two components that we had in our previous analysis? And luckily, there was a component that resembled our other cortical component that we found in the control group. However, when we plot those, we see actually there's almost no change. They're almost identical between placebo and oxytocin. And again, we see that whatever change there is, it's very heterogeneous in this population. So this is roughly where we're at now, and we kind of want to sort of tease apart this heterogeneity much further to see, like, why are there certain individuals with autism that do show an improvement and why are there others that don't show an improvement. So as you saw, and like perhaps sort of briefly glanced at in the design of the study, we've also collected saliva samples. We've done quite deep phenotyping on these individuals. So basically, this is where I'm at this moment. Where do we go from here? We're sort of planning to like go deeper into this analysis, see if there's any differences in the sort of baseline oxytocinergic system in these individuals, looking at either plasma or saliva concentrations of oxytocin. We can have a debate about which one's better later on, perhaps. We also kind of interested to see, like, is there an interaction with other hormones? Like, we had these were all women with autism, and we obviously controlled this as best we could for like menstrual cycle effects or interactions with other hormones, but there might still be some sort of residual interaction that we haven't accounted for. And then the other thing I didn't show here is like, and that's sort of been a key message I think in oxytocin literature, is we know it's very context and person dependent. So maybe there's a modulation in the autism group, but it's very sort of dependent on the specific cognitive domain that you're looking at. Because based on that first sort of neurotypical control study, you see things happening in a reward network. So maybe if you have a reward task that sort of more strongly triggers this network, that's maybe where you actually pick up effects. And then again, you can see, are there individuals in those tasks that respond more atypical than others? And maybe that's what's driving this heterogeneity. So the last slide, just want to thank a lot of people that helped me out. Especially Simon and Jack for just letting me do all of this stuff in on my ideas that weren't founded at the time, and Sarah Crockford, who spent many hours with me on the scanner, scanning all these individuals. That's it. Thank you for your attention.